This is critical thinking in growth, development, and genetics. And here are the questions. So here's the first one, where you have to identify the false statement. Here you have to identify the true statement. And here is a clinical problem. Let's look at this first question, which asks you to identify the false statement, and let's look at the statements. So here it says, normally only one secondary oocyte is released into the peritoneal cavity. Now this is true. At ovulation, normally only one secondary oocyte is released, and it is released into the peritoneal cavity. Occasionally, only occasionally, you may have two released, and if two oocytes are released and they are fertilized by two different sperms, you get what are known as fraternal twins. And hence those twins need not necessarily be of the same sex, but this is only occasionally because each ovary ovulates alternately. Let's look at the next one, and which says both the chromosome number and the amount of DNA in a sperm or an ovum is half that of a cell elsewhere in the body. Now this again is true, and here notice I mentioned the word ovum, which in the females the secondary oocyte becomes an ovum only if it is fertilized, and so it is the ovum which has half the chromosome number and half the DNA, and the sperm of course also has half the chromosome number and half the DNA. And I'd like to show you an image. And here let us look at this picture where we're looking at gametogenesis. So it is spermatogenesis in the male and oogenesis in the female. So we begin with the spermatogonium or the oogonium, and that grows or enlarges and becomes the primary spermatocyte, which you see here. There's only enlargement, it just grows in size, and the counterpart of the primary spermatocyte is the primary oocyte. So this is like a regular cell which has 46 chromosomes, and at this point it has a normal cell, let's say, has 2N DNA as is shown here. When it becomes a primary spermatocyte or a primary oocyte, at that time the only change that occurs is that the DNA duplicates so that there can be shuffling of genes. So there is, let's say, it duplicates from 2N to 4N. So here also you can see in a primary oocyte I've shown you that it is 4N. Now in order to provide gametes, which are sperms and ova, you have to make the number half. The chromosome number from 46 has to become 23, and the DNA, originally it was 2N, that's that of a regular cell. So the DNA has to become N. Remember, 4N is not regular, it has duplicated. So this is what we are aiming for. So let's look at the process here. So the first meiotic division from primary spermatocyte, it becomes what is known as a secondary spermatocyte, and from a primary oocyte, this is also a primary oocyte, it becomes a secondary oocyte. So at this point everything becomes half. So 46 chromosomes becomes 23 chromosomes, or 22 plus X and 22 plus Y in the case of the sperms, and 22 plus X in the case of the secondary oocyte, and the DNA from 4N becomes 2N. So this is what is seen in a secondary spermatocyte and in a secondary oocyte. So you have 23 chromosomes, of which one is a sex chromosome, and 2N DNA. Now in the female, the secondary oocyte, only if it is fertilized will it continue, the process of oogenesis will continue. Otherwise this secondary oocyte is just, this is the one that is liberated into the peritoneal cavity at ovulation. If it's not fertilized, it just passes out of the body. In the males, however, the process continues. So the secondary spermatocyte then forms spermatids. Now in spermatids, the chromosome number remains the same, but the DNA now becomes half. So you can see that the DNA is half, which is what we aim to get. And then these spermatids undergo spermiogenesis, which is just a morphological change, and they become sperms. So sperms therefore also have 23 plus N. Now, should this secondary oocyte be fertilized, what will happen is that this will form an ovum, and in the ovum, so you can see it's called a mature oocyte and ovum, so you'll have 23 chromosomes and the DNA will become half. The chromosomes don't half at this time, it's only the DNA which becomes half, because this second meiotic division is very similar to mitosis. And then of course you have 3 polar bodies. So you can see that's how it happens. And I just want to show you another image which is really nice and should help you. So here you can see meiosis in the female. You begin with the oogonium, which enlarges and becomes a primary oocyte, and then you can see the DNA duplicate. So here, let's say there were 46 chromosomes and 2 nDNA. Now you can see the threads are duplicating, so there's 46 chromosomes and 4 nDNA. This then divides, so this is a primary oocyte, so it divides and becomes the secondary oocyte, which will have 23 chromosomes, and you can see 2 nDNA, and it forms one polar body. Each one of them divides, and so you get 4 at the end. So then this is the ovum, which will have 23 chromosomes and nDNA. You can see the DNA has become half of this. There were 4 here, so you can see the half, and then you have the 3 polar bodies. So to come back to this statement, so this statement says both the chromosome number and the amount of DNA in a sperm and an ovum is half that of a cell elsewhere in the body. So this is also true. The third statement, which is Hcg production, human chorionic gonadotropin production, which is produced by the placenta, increases in situations where the placenta is large. This is also true because Hcg production increases where there are twins. If there are twins, obviously the placental size will be large. It increases in conditions which are known as hydatiform mole, which where the placenta sort of really enlarges and becomes like small vesicles. In diabetes also, because diabetic when the mother is diabetic, she tends to have large babies. So diabetic mothers, when the mother is diabetic, she tends to have large babies and the placenta is very large. On the other hand, you'll notice in ectopic pregnancy the Hcg production will be small because the placenta is very small, especially since ectopic pregnancies usually occur in the fallopian tube, so there's not enough space. Let's look at this fourth statement, which says gamete intrafallopian transfer, and this is called gift technique, is a method of in vitro fertilization. Now, in vitro fertilization means where there is fertilization outside the body. Now, in gamete intrafallopian transfer, read the words carefully. It says gametes are transferred into the fallopian tube. So you have the sperms and you have an ovum. Both are placed inside the fallopian tubes. And when the sperm fertilizes the ovum, the fertilization therefore occurs in the fallopian tube. So therefore, this is not a method of in vitro fertilization. So this is the false statement, and hence the answer to this question. But let's look at the last one, which says skin color is expressed through multiple alleles, and this is true. That's why you have the ranges of skin color. You know, when people of different skin colors marry, the children may be of varied color. The skin color may be varied, and that's because skin color is expressed through multiple alleles.